quasi-species. I want to give you a little bit more information that you can use to generally make answers better. So here's a nice little outline that actually comes from this paper over here of the big concepts in quasi-species theory. So you got this cloud of mutants, and then you've got an error threshold, like a point where the cloud of mutants becomes less fit if they have more errors than this, or they become more fit if they have, fewer or are close to that error threshold anyway. And then some of the things that follow out from that. So that's a nice way to kind of organize this, I think. All right, but let's talk about individual populations. So here we're showing one way to model a fitness peak. So you can model it as a single peak, and this may or may not reflect reality, but it's easy to model, which is nice. So if you have a virus and the virus has a very narrow fitness peak like this, and there's only one fitness peak, then if you have a virus that starts off near the top and it undergoes mutation, you'll end up with a population that is essentially down here at the bottom. Because remember, making a mutation is like moving in some direction. And if you're basically on this sort of pin-shaped peak, any direction you would move, think forward, backward, left, right, is going to lead you way down the slope. And so more mutations push you further from the center, which means you basically drop down to here, all these little positions on the floor. So in a situation where there are low mutations, you would expect fairly narrow quasi-species up here at the top. In situations where there are high mutations, or maybe high selection is another way to think of that, then you'll get a lot of competition. You'll get a lot of dead genomes, but the ones that survive will be up here near the top. So let's look at this another way, and I think this is nice because it has both viroids and quasi-species in it. And so it's useful for both of those aspects. So Darwin, the idea that we take from Darwin is survival of the fittest. Basically the one that reproduces the most over the short or long term is going to be the one that is favored by natural selection. Basically, they're buying more lottery tickets, and so they have a greater chance of winning because there are more of their genetic offspring. However, at high mutation rates, the ones that are fittest, the ones that can produce the most children, are not necessarily in the best situation. Because there, it's going to depend. If mutations are inevitable, and if you're in a situation where mutations are going to happen or you're going to select against the original, then having a flat quasi-species, sort of a dome-like quasi-species, could really help, which just means a situation where you can have a change in genetics, you can move away from that peak, but because the overall fitness landscape is sort of flat, like a little bubble, you don't lose very much fitness with each genetic change. This is the idea. And so that concept is again called survival of the flattest. And this is what we talked about earlier. And so this is actually an experiment we're going to go through that actually demonstrates survival of the flattest under certain conditions. And it does it with viroids. And so you remember viroids are just circles of RNA that fold up. They have a nice, strong secondary structure. And they're going to be copied by the host cell. And then they're going to contain a ribozyme inside them that is either going to cut apart the RNA or join the RNA together after the cell cuts it apart. So the two they're studying both infect chrysanthemum flowers. So here's a lovely picture of some chrysanthemums. Aren't they great? And the two viroids that they're using here that infect chrysanthemums are chrysanthemum stunt viroid, CSVd, and chrysanthemum chlorotic model viroid. And so that's CChMVd. And they're, the last word in there is part of how they're classified, but that's not really relevant to what we're doing. So think chrysanthemum stunt and chrysanthemum chlorotic model. If you put these two in together, chrysanthemum stunt is the dominant one. So we think it has a very high, very narrow quasi-species. Like if we trace it out with a mouse, it should look about like that. So that's the CSVd. Very high peak, but very narrow peak. CChMVd, so chrysanthemum chlorotic model viroid, we think has a lower but flatter fitness landscape. And so here is some of the data about this and sort of what happens. And yeah, interesting experiment. So, number one, they put these both into the same plants. What did they find? They got lots of copies of CSVd because this is the one that is narrow but has a high peak, so it grows a lot. So lots of copies of that, only a few copies of chlorotic mottle viroid. And so the ratio of these was about 20 to 1 or 0.05 to 1 if you're going this way. So if you're talking about just fitness in terms of growth, then CSVd is the fittest of the two. It produces more in a mixed competitive situation. But if you look further at this and you sequence all of the variants in there, what you find is that within CSVd, even though it produced a lot of individuals, those individuals were not very diverse. So our diversity number here is 2.47, average number of mutations per individual. And over here for chlorotic model, it's 7.7. So it's about 3 times as high. So there are 3 more mutations per genome in chlorotic model compared to stunt, which is CSVd. And so that's interesting. So we've got a broader quasi-species, but it doesn't grow as well. That's our kind of low, sort of flat fitness landscape. And then robustness. I think this is the interesting one. So they're looking at, since the viroid is just a circle of RNA, the 2 things that it has are a genetic sequence, and so we covered that in the diversity section, and it has an RNA secondary structure, so it folds up. The optimum RNA secondary structure is if basically everything on the one side is paired to everything on the other side, so it's just zipped together, and that way it can squeeze through little pores between cells, for example. And so, yeah, the more base paired it is, generally the better for viroid fitness. And if you look at CSVd, it has a very narrow deviation, so it doesn't change much. Basically, it's always pretty close to optimal secondary structure. So everything on the one side is mostly paired to everything on the other side. Compare that to chrysanthemum chlorotic model, and look at that, 81. And so it's 27 times more likely, or it's going to have 27 times more deviation from that perfect, that ideal of the 2 sides being complementary to each other. That's really something. That is pretty astounding. So this is what tells us that even though this population is more diverse, and that's this number up here, it also is able to tolerate that diversity and diversity of shape and diversity of sequence. So that's interesting. But overall, it doesn't grow anywhere near as well as CSVd. That's just the one that is fitter. So then they did the actual experiment, and this is the coolest part. So ultraviolet radiation can cause mutations. And ultraviolet A is kind of the least likely to cause mutations, but the shorter wavelengths, things like ultraviolet C, this is what they called hard ultraviolet. This is really likely to cause mutations. And so they exposed the plants with these viroids to a lot of UVC or to no UVC. They used different light bulbs for growth of the plant with the viroid inside it. And the idea was that this UV is going to cause mutations in the viroid because it'll, modify the chemical structure of the viroid. That's what UV does. And so they then looked at what does this do to the relative percentage of the 2. So chlorotic model is always very low compared to chrysanthemum stunt, CSVd. But as you increase the amount of UV for weeks and weeks and weeks, look at that. Chlorotic model starts to do better relative to CSVd. So it has a selective advantage under high ultraviolet conditions. And we think this is because it's able to tolerate a wider range of mutations that change both its sequence and its secondary structure. Compare the two, though, under the presence of no ultraviolet, so there's no mutations coming in, and CSVd just outcompetes chlorotic model to the point where chlorotic model is headed for extinction after about 6 weeks. Probably somewhere around 8 weeks is when we'd expect that to actually become extinct. And this is because in the plant there's a limited number of polymerase II, limited number of cells to infect, and so these two viroids are actually competing against each other for scarce resources. And so, yeah, under different conditions, when there's high mutagenesis, the one with the flatter quasi-species actually has an advantage, and that's why the line goes up. And under circumstances where there is very little mutation going on, so little reason why it pays to be broad, then the one that grows best is the one that's going to predominate. And the one that's broad but low is just too slow. Yeah, it's basically the tortoise and the hare. Think of CSVd as the hare. Think of CChMVd as the tortoise. And here's what happens to the average nucleotide differences. So this is without UVC, and this is with. So the arrow shows going from without to with. And so this is the diversity number. So over here is more diversity, over here is less diversity, and same thing with the up and down. So basically, numbers that are up here, those are highly diverse. Numbers that are close to this are have very little diversity in their genome. And so before UV, CSVd was able to tolerate actually a fairly wide range of mutations. After you put in that ultraviolet, though, bam, it gets crushed down, and there's basically only a very narrow quasi-species that's able to survive. So there was a very limited number of solutions that could still be viable when there was a high amount of ultraviolet there, when mutations were basically forced. Compare that to chrysanthemum chlorotic model, and this is its position before, so pretty high diversity. And this is its position after UVC. Basically, it goes up in diversity when you apply the ultraviolet light. So it's making these mutations. The mutations are being selected out for CSVd, so they all just go off and die. But for CChMVd, those mutations are actually tolerated, and so they accumulate. And so you end up with more and more different kinds of virus for chrysanthemum chlorotic model in this situation. So basically it looks like this. If you imagine this is CSVd without UVC, when you turn on the ultraviolet, because it's a very narrow single peak, most of the viroids end up dead. And a few of them are sort of clinging to the low fitness end of the spectrum because you put all those mutations in there, and it's a very narrow quasi-species. Compare that to chrysanthemum chlorotic model. Without UVC, it's a pretty lackluster thing. It's always pretty close to death. But with UVC, it's still forced to mutate to exactly the same extent as up here. But because the quasi-species is flatter, the ones that are near the top actually pay a relatively small price for that compared to the price paid by CSVd. And so they actually do better overall. They sort of outcompete CSVd under those circumstances. So isn't that neat? It's a viroid, and it is comparing a flat quasi-species with a very pointy quasi-species. And this is actually a situation where under UVC, ultraviolet C selection, the flatter quasi-species actually does win. Survival of the flattest, demonstrated right there. And so, this is both a quasi-species concept. Survival of the flattest is definitely one of those. And since we're doing it with viroids, it's a viroid concept and would be very good in a viroid discussion. So there you go. I hope that helped. Thanks very much.